In this lecture, we're going to discuss zooarchaeology, which is a specialist among archaeologists. Zooarchaeology is the study of faunal assemblages or animal remains that are recovered from archaeological sites. And it's important to keep in mind that faunal assemblages differ from paleontological assemblages because we assume that humans played some sort of role in their formation. The faunal analysis itself involves identifying and interpreting the faunal remains. So this involves first assigning specimens or particular bones to an element, that is a certain part of the body, classifying specimens to taxonomic categories, so depending on how much is present, you can identify the species, genus, family, or even the order. Sometimes, however, only a size class can be assigned to a particular specimen. So small, large, or a medium or large mammal, for example, just general categories of fish versus reptiles, things of that nature. So zooarchaeologists are specialists who have an understanding of animal anatomy, but they also rely on comparative collections that contain modern fauna of both sexes and different ages that help them to make identifications among archaeofauna. When a faunal analysis occurs, there's two primary forms of data that should come out of any kind of analysis. The first being the number of identified specimens, or NISP, and this represents the raw number of identified bones or specimens per species. So that's what you're seeing represented in the table here. Clearly a lot more bison bones in this particular assemblage than any other species that has been observed. The other form of data then is called the minimum number of individuals, or MNI, and in this case it's the smallest number of individuals necessary to account for all of the identified bones. So it's very different in that case from an ISP. How you go about calculating your MNI then is you tabulate the bone frequency by element. So you separate left femurs into one pile, right tibias into another one, the hyoid bone or any kind of cranial fragments into another. You assign specimens to the correct side or element of, the correct element and the correct side from that species. And then you compare fragments to see whether two fragments are from the same bone or if they actually represent two individuals. So in the case of the coyote bones that we see here in the table, if all three bones are left femurs, for example, well, we know we have three coyotes represented in that assemblage, whereas the bison, there's 1,000 NISP there, but might only be one or two individuals itself. It's going to depend on the analyst and what they're able to observe. There are also important modifications for the zooarchaeologist to note. Natural modifications include gnawing by carnivores, which is what we're seeing in the photo to the right here, or rodents themselves getting into trash pits. Human marks are obviously going to be important because we want to know how humans were modifying and interacting with the bones themselves. So any kind of cut marks, flakes, the spiral fracture that you see in the photo here maybe would have helped for extracting the bone marrow, or if the bones themselves become worked into a tool of some sort, perhaps. Also, what we're seeing in this photo then are different degrees of burning that might have taken place, that might have occurred or might have impacted the state of the bone before it entered into the archaeological record. And so those that have been exposed to very high heat or long heat become very white in the end. Now your textbook talks about a particular example that is quite interesting, and that is a faunal analysis that occurred at the site of Chavin de Huantar in South America. This is a ceremonial site that dates to 2850 to 2200 BP. And what the faunal analyst was able to interpret, so you see the results of their analysis here. As we go up through time, we're seeing this increasing dependence on camelid species, while the wild animals, the deer that are in the area, they become less dependent on. And so your textbook talks about charqui, I'm not sure how to pronounce that, which is actually a Quechua term for freeze-dried llama and alpaca meat. So what the archaeologists were able to learn in this case is that the early occupants used llama mostly as pack animals, but camelid meat was a byproduct of culling their herds over time. So there was an increasing trade in this jerky that allowed them to trade jerky out and get agricultural products in, or at least that's the interpretations they were able to make. So quite an interesting case, and something interesting that we're able to learn about a past society through a faunal analysis.